cannot help but think that Allah subhanahu wa ta'ala has made our bodies in such a beautiful way that He kept the treatments of all these diseases inside our own bodies. And He just left it for the mankind to discover, and we're slowly moving towards that. To elaborate on the exosomes, I would like to invite again Professor Faridoon. Sir, please.
I promised Bushra I won't cross 15 minutes. And second, it gives me a great pride that mashallah jo hai, about maybe more than 50% of the speakers today are my former students. Dr. Bushra, Dr. Mustasan, Rifat is also here in Ghazala. Third, besides doing all that, I still haven't been able to convince my son Jawad to opt for biology. So anyways, I'm going to talk about exosomes, and basically what I will be talking about is not all exosomes are created equally. So exosomes, where do exosomes come from? They come from the cells which are present in our body. As a matter of fact, blood is the blood we have in our bodies. They have more than 1 trillion exosomes. It's a natural phenomena. Cells, all eukaryotic cells, release these small vesicles. Exact physiological function is not known. It is thought that they are part of garbage disposal system. Thank you. Or it could be also a mechanism for cells to communicate with each other. So we know about signaling molecules that signal cells to perform their function for coordination between different cells. We know there are different hormones and other signaling molecules, but more significantly, a more significant effect can be generated by exosomes. So, anyways, all cells, as I said, create exosomes, and our bodies are full of exosomes. Most secretions, saliva, blood, milk, they all have exosomes, and they're created for a purpose that we will discuss further in our presentation. So first of all, I'm going to talk about how the exosomes are generated, what do they carry inside them, and how we can actually manipulate different exosomes for a specific purpose. And also, I know there are a lot of questions. It's a hot burning topic. Everyone wants to know about exosomes and want to use exosomes. They're already in the market. And also, I'll give you a specific example. An institution in US recently contacted me regarding hereditary spastic paraplegia. It's a very, very complicated disease. So one of the strategies I proposed was using exosomes that are engineered. So we will talk all about that. First of all, how exosomes are created. Cells generate different type of vesicles. Some of them are pinched off from the plasma membrane. Those are called ectosomes, not exosomes. Here you can see a cell is pinching off a little vesicle. Ectosomes are slightly larger than exosomes. They range in size from about 50 nanometer to 1 micron. Exosomes, they are actually generated by endosomes. Endosomes are small organelles that are part of the endomembrane system. So they receive packages from Golgi apparatus and also whatever is endocytose. So they combine, and also proteins that are formed in the cytoplasm, they are also pumped into the endosomes, and there they can be packaged. So in the endosomes, some endosomes mature into multivesicular bodies. So these endosomes have many, many small vesicles inside them. So when these vesicles fuse with the plasma membrane, as you can see here, the smaller vesicles are released extracellularly. And these vesicles then again perform their function, either getting rid of extra material, membrane, or other material that cells have accumulated. And also as a communication devices, to communicate, to coordinate the cellular efforts in an organism. The contents of an exosome. So exosomes, they have some proteins on their membrane which are in a much greater number. For example, tetraspanins, you can see CD81, CD9 and CD63, they are actually called tetraspanins, a group of proteins that decorate the membrane. They have many other transmembrane proteins. They have DNA in them. They have RNAs, microRNAs, non-coding RNAs. Of course, they have lipid membranes, so they are carrying a lot of lipid component. They also have cholesterol in the membrane. So this is basically what an anatomy of an exosome looks like. Exosomes, what are the functions? Some of the functions exosomes can regulate or influence: regulation of gene transcription and translation, survival and proliferation of other cells, they can influence that, angiogenesis, wound healing, waste management that I mentioned. Also they have a profound impact also on the immune system. Exosomes, they can fuse with the other cells based on membrane interaction, but you can also specify which exosome is going to interact with which cell type. We will talk about that, how you can engineer exosomes that will have only, they will deliver their cargo only to a specific type of cells. So why does it, I say not all exosomes are created equal. It depends upon many factors. One of the most important factors is what is the parent cell. We have hundreds of different types of cells in our body. We have heart cells, we have kidney cells, we have muscle cells. They're all producing exosomes. The function of these cells is different. Since they're performing different function, their components are also different. The cells, when they generate an exosome, they're basically packaging many of the things, many of the contents that that particular cell is making into these small vesicles and releasing them in the media. So, as I mentioned earlier, blood has about 1 billion per ml, 1 billion exosomes per ml of your blood, which roughly comes to about 1 trillion exosomes. So why are we giving exosomes using exosomes as a therapeutic tool when you already have more than 1 trillion in your blood? So we will talk about that too. So other factors that influence exosomes is if you're collecting exosomes from cultured cells, so what type of media you're using, how you are growing cells. So you can isolate exosomes either from the natural fluids, you can isolate exosomes from the blood. Depends upon how you are preparing your exosomes, whether you are using naturally occurring body fluid, or are you getting your exosomes from cultured cells. So that is one of the things. Also you can process exosomes through different techniques. Either you can use ultracentrifugation that produces better yield as compared to the kits. There are kits available in market that you can use for both, either if you want to prepare exosomes from blood or from the cultured cells. You can use kits or either use the kits or ultracentrifugation method. The other factor that influences how these exosomes are going to perform is whether they are natural exosomes, whether they are collected from the media that cells are living in, or whether they are engineered. So I will briefly talk. I purposely did not want to put too much technical details in this presentation, because I know most of us here are clinicians, so that could have been a little too boring. Cargo loading. So if you want to deliver specific material to specific target tissue, you can actually take exosomes and you can package different material into it. I'll give you an example of a specific disease, as I mentioned, hereditary spastic paraplegia, and you need specific proteins in those exosomes to produce a clinical effect. So sometimes what you do is actually, if you want to package proteins that are that want to stay away from aqueous material, you can actually add those proteins into the media, and exosomes will passively acquire them. They will just find, if they're hydrophobic material, that was the word I was looking for, hydrophobic compounds can enter the exosomes on their own. You can make this process several times, an order of magnitude greater, by using other techniques, but exosomes can take drugs if you incubate exosomes with that particular drug. Either you can incubate exosomes in that drug, or you can incubate the parental cells that are going to generate that exosomes. You can also treat the cells, the parental cells, with that particular drug. Another technique which is used is sonification with the homogenizer. You can do freeze-thaw cycles. Either you can freeze the exosomes, drop the temperature to -80, or in liquid nitrogen, if you cycle, put the compound in the media in which your exosomes are present, and if you go through freeze and thaw cycles, these exosomes will be more amenable to taking up that particular agent, therapeutic agent. RNAs, if you want to use a specific construct of RNA, you want to instruct cells to produce, your target cells to produce certain protein, you would need to put messenger RNA into these exosomes. It works better if you used electroporation device to load your exosomes with that particular messenger RNA. There are also certain membrane polymerizers. For example, saponin, if you add your material that you want to load into the cells, and if you add saponin, the take-up is 10 times faster than passive take-up, which I mentioned, which was the first technique I mentioned right here. So again, if you want to look at more details about how you can engineer the internal cargo of a particular exosome, you can look at this paper. It's a very nice paper. So initially when I said that why we need more exosomes, so exosomes, naturally occurring exosomes which are already present in your body, they will not have any of this material. So when you're thinking about targeting a particular disease, you have to have special exosomes. Not only their cargo should be specific for your particular disease, but also they should be targeted. So do we know any mechanism how certain entities can go to specific cells? Okay, for example, viruses. Viruses have certain proteins on their surface that allow them to attach to specific cell and deliver their cargo there. You know many examples. For example, HIV, it has profound affinity for CD8 positive cells. Your helper T cells, CD4 positive cells, helper T cells. So you can use different strategies. Nature has done many experiments for us already. So if you incorporate a particular protein into the exosomal membrane, you can use that as a targeting material. All our cells have many receptors on their surface. You can also decorate the exosome with a specific antibody that will go and find your target cell and only hit those cells that you need to manipulate. You don't want to deliver your cargo to each and every cell. We'll look at an example when we discuss HSP. So most commonly asked questions about exosomes is, how long do they circulate? So you have injected someone with an exosome material, how long will it last in the body circulation? So the answer is about an hour. They don't live in your circulation too terribly long. Now, if you remember, I told you that exosomes are one of the functions is to communicate. the coordinate cellular efforts. So the purpose is that when cells are releasing exosome is not for them to hang out in the blood or body fluids, they have to go and get absorbed in their target cell or other cells. So exosomes are taken up very, very quickly by the other cells. So if you add them after an hour, they'll be gone. But their effect lasts much longer. So how long the effect lasts? Generally, again, it's an emerging field. So the current data suggests that their effect lasts about 6 to 8 months. So after that, you have to again supply exosomes to your patient. Either you could do that, or you could use the parental cells that you have modified. So once they are in the body, they'll continually produce those exosomes, and you won't have to administer exosomes repeatedly. So initially you give exosomes to see the effect, whether they're causing the same effect that you want in your patient. Once you see that effect, then rather than using exosomes, injecting the exosomes over and over again, you can give a more permanent solution to your patient by injecting the parental cells. So what is the dose for patients? Now this is very important because exosomes are being made available commercially. So you need, when we talk about stem cells you're talking about in millions. You need about, I would say, 60 million to 50 million nucleated cells from SCF to cause an effect. So if it is anything less than that, you are not going to get a desired effect. So when we are talking about exosomes, the figure is in billions. It's not in millions, it is in billions. Okay, so they are nano-sized particles, about 30 to 100 micron in diameter. So if you want to use exosomes and you want to see an effect, you have to ensure that you are using sufficient quantity of exosomes, otherwise they will not produce the effect. So anyways, here is the reference. So you can look at it. So currently, these are the clinical trials that people are which are currently going on, and this is the total dosage. I know the print is very small, but I had shortage of time, so I could not make a bigger picture of this. So again, this is in billions. So you need billions of exosomes for particular to treat a particular disease. And when we are talking about cells, if you are going to inject parental cells, how many of those parental cells do you need inside the body or in the media to produce the desired number of exosomes? So it is thought that one cell will produce about 1,000 extracellular vesicles or exosomes. So if you use that formula to come up with a billion number, you need about 100 to 1,000 million mesenchymal stem cells to produce that amount of exosomes. So, and if you have anything less than that, you will not be able to acquire that. If you do not acquire that particular number, you will not be able to get the desired effect. So we are, I think, doing fine on the time. HSP, hereditary spastic paraplegia. So spastin is a gene that has that does many functions inside the cell. It decorates microtubules, it can sever microtubules, and the M1 form of this protein basically restricts the amount of lipid a cell is producing. So in neurons, if this protein is mutated, it will result in overproduction of these lipid droplets which produce lipotoxicity. So it will kill those cells, and these patients lose their mobility, they are wheelchair bound, they cannot move. So we have two problems. Dominant negative effect, that the mutated protein is acting as a dominant negative. It is producing the bad protein, the mutated protein is hurting the cells. We need to get rid of that protein. We have to stop the production of that protein, and whatever protein is already in the cell, we have to get rid of it. Two things. Number two, haploinsufficiency. You need two copies of this gene to produce enough protein for cells to function normally. So if one of the genes is mutated, you only have half the amount of normal protein there. So how do you treat a disease like this? You have to get rid of the protein, the defective protein that is already present in the cells. You have to stop the production of that particular protein, and also you have to come up with a solution that you are producing enough normal protein inside those cells. So this is very tricky. You cannot fix this problem with stem cells. There is no type of stem cell that can clean out the cells, get rid of the protein, stop the production of defective protein. So you have to go back to the drawing board and try to fix this problem through some different technique. So this is what I propose to them. One of the things you do is you prepare exosomes with antisense oligonucleotides. Antisense oligonucleotides, what they can do is they can prevent production of more defective protein. That is how antisense oligonucleotides work. So I'm not going to again go into detail of how they function, but that was one of the things that you could do. The second thing you did is what I proposed. It also, if you produce a vector that is producing two messenger RNAs, one is for normal protein, other is to inhibit the abnormal gene. It is going to inhibit the abnormal gene from functioning and produce the normal protein product. So what do you do with the defective protein that is already inside the cells? You cannot get rid of that protein with any type of stem cell therapy. So one of the techniques what you can do is actually you can conjugate ubiquitin ligase with a receptor for that particular protein. So the receptor has affinity for mutated spastin, and ubiquitin ligase will put small ubiquitin molecules on that particular protein. Any protein that has ubiquitin attached to it goes to proteasome and gets destroyed. So this was the strategy that I proposed to that institution. They are working on it. So the basic point is you can use exosome as a device for delivering a specific reagent or therapeutic agent to a specific cell type. You are hitting, it's like more like a smart bomb, a laser guided bomb that will go and hit the target and deliver its contents and fix that particular problem. So basically, I have already discussed what I proposed to them with you. The way forward. Exosomes should be from the right source. From the right source should be used. So you don't. By the way, this presentation is going to be available if someone wants to take it. If someone wants to photograph, that's fine too. Exosome from the right type of cell. The source of the cell is very, very important. I said we have hundreds of different types of cells. If you are going to use naturally occurring exosomes, then you should culture the right type of cell and extract exosomes from that. Appropriate number should be used. So one of the problems with failure of stem cell therapy is lack of appropriate number of stem cells that you're using as a therapeutic agent. So that is one of the top reasons why stem cell therapies fail, because you are not getting enough stem cells. Right type of exosome should be used. So if you are going to treat something like hereditary HSP, hereditary spastic paraplegia, and you are using naturally produced exosomes, it is going to do absolutely no good. So if a disease that you want to cure or treat requires specially engineered exosomes, you have to use those specifically engineered exosomes. Appropriate route should be used. Actually, this is less of a problem for exosomes because they can cross almost any barrier. The hardest barrier to cross is blood-brain barrier. So there was a paper on one of the previous slides I forgot to mention. Exosomes can cross the blood-brain barrier. So initially they were institution I talked about in US, they were going inside the brain and injecting their reagent there. You don't have to do that. Even if you inject exosomes in the blood, they will go to the brain. So, duration of effective period should meet expectations. So, you cannot market your exosomes as a permanent fixture. They are not permanent fixtures. They get absorbed. Their effect lasts several months, but after that you'll have to give another dose of exosomes. That is basically, I know, I had promised 15 minutes nahi ho gaye. So the other thing I wanted to mention is Bushra, myself and Dr. Mahmood, we are also producing our own exosomes, primarily for our purposes, but if someone else is interested, they can contact Dr. Bushra. So if there are any questions regarding exosomes, I'll be happy to entertain them.